Now we'll see a very interesting topic, the growth and nutrition of bacteria. You know the factors that affect your growth and development? Yes, they can be heredity, growth hormones, or nutrition, or environment, or your requirements, or basic requirements like food, air, water to be alive. Yes, any of these or all of these? Yes, all of these are required for the growth and development of any individual, the human being. Okay, how will then the bacteria grow? Okay, they can be like they can synthesize their own food. Okay, the metabolism can happen by themselves. They need not depend on others. They are said to be autotrophs. The other one, the heterotrophs, or many of the pathogenic bacteria, means the harmful causing bacteria, okay, or heterotrophs, or it can be like parasitic in nature, okay. They depend on the host, means the other cells, for their metabolism. So they are said to be heterotrophs. And the basic nutritional requirements that are essential are hydrogen and oxygen, carbon, nitrogen for ATP synthesis. All these basic requirements, including the minerals, organic growth factors like vitamins, nicotinic acid, folic acid, vitamins like B12, all these are the essential nutritional factors that is required for the growth of bacteria. Then there are some environmental factors too, like water, oxygen, moisture. Of course, it is needed. 80% of bacterial cell is made up of water, and based on the oxygen requirement, the organism can be the bacteria can be divided as aerobic, anaerobic, aerotolerant, and facultative anaerobes likewise. Then based on the carbon dioxide also, they some organism require at least 5% carbon dioxide to grow. Anaerobic organism, they require most of the carbon dioxide and nitrogen, no need of oxygen. So depending on their requirement, carbon dioxide is also needed. If carbon dioxide is likely for the growth of the bacteria, then the bacteria is said to be capnophilic bacteria. Then temperature is also required. The optimum temperature for the growth of bacteria is 37 degrees Celsius, and it can be like psychrophiles, mesophiles, and thermophiles. Psychrophiles are the one which is grown in the temperature less than 26 degrees Celsius, and the mesophilic bacteria are the one which have the optimum temperature like 37 degrees Celsius, and it can be plus or minus like 26 degrees Celsius to 40 degrees Celsius. And the next is the thermophilic bacteria, which are like they love more temperature for its growth, like 60 degrees Celsius to 80 degrees Celsius. Next, pH, the hydrogen ion concentration, that is also required. The optimum pH for the bacteria to grow is 7.4 to 7.6. Okay, if they require high pH, then they are said to be halophilic organism. Then light is also required. They are said to be photochromogenic organisms. Osmotic pressure, of course, is very important, and symbiotic antagonistic activities are also important. Okay, so these are all the basic needs of the organism to grow. Then coming to the bacterial metabolism, they can be like aerobic organism, aerobic respirators, otherwise said to be oxidizers, anaerobic respiration, otherwise said to be fermentation, and facultative anaerobes to undergo the anabolic or catabolic reaction. Anabolism and catabolism are nothing but the metabolic reactions, the building up process and the breaking down process. And depending on the aerobic respiration, they can undergo oxidative phosphorylation for the ATP synthesis. If they are anaerobic in nature, then it will be like a substrate level phosphorylation. Okay. How do the bacteria multiply? So what are all the requirements we have seen? How do they multiply? Eukaryotes will be dividing based on the mitotic and meiotic cell division. Bacteria, the prokaryotes, usually multiplies by a binary division. Okay. First will be the the cell wall and plasma membrane will be starting dividing, and the nucleic acid is important. First division starts with the nucleic acid. Then the parent cell will elongate. The elongation process takes place. The cell envelope will grow inwards for the division. Formation of a transverse wall of the cell wall will appear, and then the cells will get separated. This is how the binary fission takes place. Next, coming to the bacterial growth cycle. Okay, we can measure the bacterial growth when a suitable culture medium is inoculated with a bacterium and incubation is going on. Its growth leads to an increase in the number of bacterial cells, which follows a definitive course. Yes, it will be logarithmic in phase. While this time we are supposed to know the generation time of a bacteria. The optimum generation time of a bacteria is 20 minutes. Okay, one bacterium to divide into 2 means at the end of binary fission we will get 2 daughter cells. That time period is said to be generation time. The generation time for a Mycobacterium tuberculosis is 20 hours, and for Mycobacterium leprae it's 20 days. So that is why Mycobacterium leprae is tough to grow in the artificial culture medium. So when we grow like that, okay, after some period, every hour or every 2 hours, logarithmic increase in the growth can be observed in the culture medium, especially in the liquid culture medium. So we can make a plot of these OD values. The optimum density values can be calculated and it can be plotted, and we get a curve of the type which I have shown here. This is called as the growth curve, the bacterial growth curve. It has 4 main phases. First is the lag phase. Okay. Second is the log, otherwise said to be logarithmic or exponential phase. Third is the stationary phase, and then the phase of decline. The lag phase, it is a one immediately after inoculation into the culture medium. Okay, it will be calculated. Usually it will be like a 0. With increase in its size, the bacterial size and the metabolic activity, the bacterium, it will attain a critical stage, a starting point of the multiplication. So that covers the lag phase. Here we cannot see the numbers increase. Okay, we just can see the size increases. Okay, so the length of the lag phase usually depends on the type of bacterial species, the size of inoculum, the quality of the culture medium. So all these determines your lag phase of the organism. It can be as short based on your nutrition provided, it can be so long again based on the nutrition. The logarithmic phase is the one the cell division starts, then proceeds, and it will be have a logarithmic increase in the number of bacterial cells. The generation time in log phase is constant for a given bacterial strain under same set of conditions. If you can provide the nutrition constantly, then the logarithmic phase can be even. So where can we have this kind of constant supply of nutrients? That can happen in the industrial areas. In the industries for food productions or some fermentation processes, if you can constantly supply the nutrition, then the logarithmic phase can be increased, prolonged. That is said to be continual growth. Okay, batch culture is different. Then is the stationary phase. After some time, when the bacterial growth ceases, means when your nutrition is exhausted, then the accumulation of the toxic products will be there. There, your number of cells that is multiplied will be equal to the number of cells that is dead. Here only we can see some of the enzyme productions and the antibiotics and the exotoxin production, the gram variability, all these can be seen in this stationary phase. Then comes the decline phase, where the number of bacterial cells will be reducing. Live bacterial cells will be reducing. Okay, so this is said to be decline phase. So involutionary forms means the cell wall deficient forms are usually seen in this phase of decline. Okay, hope you have understood the 4 phases. The first is the lag phase, second is the log, then the stationary phase, and the phase of decline. Okay, so every time we can count both the viable count of the bacteria and also the total count of the bacteria. Total count is nothing but altogether the live and the dead bacterial cells that can be counted, that is the total count. Viable count is nothing but if you can count exactly the live bacterial cells, that is the viable count. Viable count can be usually estimated by spread plate technique or pour plate technique, gives you the viable count of the bacteria. The live bacteria can be subcultured and it can be estimated. So during this bacterial growth, there are so much of enzyme production, as I have said, that can start from the stationary phase, enzyme productions, toxins productions and all. So what are all the toxins that can be produced by the bacteria? Namely exotoxins. Usually gram positive organism produces the exotoxins, and the endotoxins, usually the lipopolysaccharide that is the present in the gram negative organism, they can produce the endotoxins. So what will be the property? It will be antigenic in nature, and it cannot be filtrated. Usually these enzymes and the toxins cannot be filtrated, so they can be seen even in any case of fluids that can be used in the intravenous and that can be used for culture techniques, all these fluids. Then we have to think about other methods how we can filter them. Okay, extracellular enzymes are also there, like collagenase, coagulase, hyaluronidase, streptokinase, hemolysins. All these enzymes can also be produced in the phase of stationary. So hope you have understood the bacterial growth phase.